Hello, good morning, and welcome to today's international seminar. Um, we have the privilege of having with us Dr. Elcio Guimaraes, who is the director of the Rice and Bean Center for Embrapa. Elcio is, I would say, a good friend of mine. We have overlapped at CIET for quite some time in Colombia. But I got to know Elcio even earlier before that. Elcio graduated from Iowa State University a month before I joined Iowa State University. And everybody in our department got to know Elcio because of the notes he left for all the plant breeding courses that we took, which were excellent. Not only the class notes, but also the exams with answers. So Elcio, we owe you a lot of former Iowa State, a lot of alumni from Iowa State from that time owe you a lot. Thank you so much. Elcio has been the director of this center, of the Embrapa Center for Rice and Beans since 2019. Before that, he was the head of research and development, the director of research and development at that institution. Before that, he was director for Latin America at CIET in Colombia. Prior to that, he spent time at FAO as a senior officer in the cereals crop breeding division. And before that, he was a rice breeder both at CIET and Embrapa. Elcio has published a lot of scientific articles, book chapters, and books. And he has released, he's participated in the release of more than 40 rice varieties, not only in Brazil, but in Latin America. Elcio, it's a pleasure to have you with us. Thank you so much for accepting the invitation. I'm going to stop sharing and I'm going to give you the floor.
Can you hear me?
Yes.
Okay. Thank you, Carlos. You were always so kind. You didn't change a bit. Your kind words regarding my person. Yeah, it was a privilege to know you all the way from Iowa State and the work we did at CIET. So thank you. And the images that you showed, the rice, make me feel back to CIET because many of them were from Colombia.
Yes,
so.
a lot of those images are from CIET. Yeah, go ahead and start sharing your screen.
Okay. Okay, there you go. You just let me know when you see it.
Yes, perfect.
Is that okay?
Yes.
All right. So let me put my pointer here. Okay, that's it. Okay, so again, Carlos, thank you for the invitation. It's a pleasure to participate in the International Plant Breeding Seminar Series. For us, rice breeders, is always a privilege to talk about rice. I know that there are some of them here. I saw Fernando Correa back there, our friend and colleague at CIET. So it's always a big pleasure to have the opportunity to talk about rice, mainly talk about a very successful story, which is the rice breeding story here at Embrapa in Brazil. And most of it is due to Colombari. Colombari is the head of the breeding program here for more than 10 years and helped me to put together this presentation. So if he is with us, thank you, Colombari, for the help you provide in the preparation of this seminar. I want to start by telling you that there will be 6 components in my talk. I will talk you a little bit about Embrapa in the beginning. Then I'm going to move to what we're breeding for, how we're doing it, some of the results, talk about seed market, and end with the partners that we have. But there are three main points here that I want you to keep in mind. Is the breeding strategy is key for the success. The other component that's key for the success is also the focus on the market demand. And the third one is how we do it in terms of partnership. So if you don't have partners, if you don't look at the demand, if you don't have good breeding methodology, it's going to be difficult to get any success. Fortunately, we have been successful because we have been looking at these three components very carefully. Let's start by talking about Embrapa. Embrapa, for you that might not know it, is the Brazilian Agricultural Research Corporation, and I'm going to call it from now on Embrapa. Is the public institution in Brazil, and it has 7 central units, which are in Brasilia. Brasilia is right here. Can you see my pointer? I guess so.
Yes, yes, you can see it.
Okay, thank you. We have 43 research units that are spread all over Brazil. As you can see, the different names here and the dots. So they are all over the country. And we have about 8,000 people working with us. About 2,000 or 2,300 are researchers, same amount of analysts, and these researchers and analysts is a division we have here. Most of the analysts do research as well. So we have about 30 to 35% of our group responsible for doing the research here at Embrapa. If we look at a little bit further in terms of the numbers, we have out of that 2,000 researchers that I mentioned before, we have 414 breeders. Most of them, 368 are plant breeders, 66 are animal breeders. Here where I'm located, we have 55 scientists or researchers, 16 breeders, and they're split half and half, 8 in rice and 8 working with beans. At Embrapa, we have 78 breeding programs that goes all the way from cereal to microorganisms. So where we are, I mentioned to you that Brasilia was right here. So we are 200 or about 150 miles south of Brasilia, right in the middle of Brazil, right where the Cerrado region or the Savannah region is, where the soybean, the cotton, the maize that we export is produced. So if we put your finger in the Brazilian map, you probably will put your finger right where Embrapa Rice and Bean Center is. Here is an overview of the unity, the Embrapa unity. And here is the sunset in our Embrapa entrance. Let's look at about rice in Brazil before we move into the breeding program. The rice history in Brazil is very interesting because at the very early 80s and 70s, we had about 6 million of hectares of rice, and most of it, about 80%, 85% was upland rice, grown in these savanna areas, in the Cerrado areas. As you can see, time went by and we dropped from 6 to about 1.7, 1.6 million. And that was basically because of the drop of the upland rice, which went down from 4 million hectares to about 300,000 hectares. But if you look at total production, it still increased. And that is about what we need in the country. We normally have a demand of 12 million tons, and this is what we have been producing since the 80s. So if you look at the Brazilian consumption and production, they match almost 100%. But what's important, and which is related to what I'm going to be talking to you in the next slides, is what you see in this picture here. A constant increase in kilograms per hectare. Productivity has been increasing since early 80s and has not stopped. So let's move to the breeding program. What are we breeding for? The main I just listed 5 components or 5 elements that we are breeding for. First one is yield potential. Farmers always want more yield, and more yield. And that's one trait that we put a lot of attention. Disease resistance. Rice has some diseases that are very important. Blast is one of them. Fernando Correa, who I mentioned before, is our reference for everything that's related to disease resistance. But blast is the main one. Grain quality. If you don't have grain quality, your material is not going to make into the market for sure. So grain quality has to be linked to the Brazilian standards, which may be different, which is different from other countries. So we have to pay a lot of attention on what we do for grain quality. Lodging resistance. When we have high yielding materials, lodging is always associated to it and may become a problem. Herbicide resistance. Since most of our rice is planted in the south under irrigation, and herbicide resistance is a must for that. This is also true for upland rice. So the 5 major components that we're breeding for are true for upland and for irrigated rice. They may vary in importance, but they are the same for both systems, cropping systems that rice is grown in Brazil. How we're doing it. And here I'm going to go into a few steps where I'm going to show you the different elements that we have in our breeding program and how we are moving through them. Let's start with our gene bank. We have a team headed by a breeder working in our gene bank. Flavio Brezeghello, the former head of the center here, is now the head of the gene bank. And there we have about 27,000 entries from more than 100 countries. And there is where we go for our sources of material for upland and as well for irrigate. So this is, keep in mind that our gene bank is being used as source, and we have wild relatives there. We have Glaberrima and different types of sativa there, which are very important for our breeding program. These steps that I'm going to go through now. There are 5 steps where we focus to generate and to make the success that I mentioned to you in my opening statement, saying that our program has been very successful, and this is the key reason for it. I want to start by focusing on this component here, the recurrent selection component. Our program has been doing recurrent selection for the last 3 decades, 2.5 decades. It's the only program in the world that uses recurrent selection as routine in the breeding, as part of our breeding strategy. We started with 5 populations for upland ages ago. We reduced to 4. For irrigated rice, we started with 5 as well. And now we're down to 2 populations. We have populations for different traits. Now they are more broad-based populations than specific populations for specific traits. And what we do here, we use male sterility for the irrigated program, but for the upland program, we do it manually. So it's a lot of work, but our capacity here to make the hand cross is still quite good and is allowing us to achieve the cycles, the recurrent selection cycles that we have been carrying on. As you can see here, we start with obviously recombining the materials that are coming out of the gene bank, and they come out of the intermediate populations where we have quite good amount of favorable alleles in the parents, and we run them through S0 plants and F1 progeny and go back circularly. So we do selection here, evaluation and selection in the progeny as S01 progenes, and we go back and recombine them and keep running in cycle here. And obviously, every time that we find good things, we take them out of the cycling and bring them to S02 and phenotype them for traits like quality, insects and disease resistance. We do have markers that we have been using in this recurrent selection process, markers mainly for BLAST. We have 3 genes for BLAST marked and 2 KTLs marked for BLAST as well that we use in our routine here. For upland, we add a marker for phosphorus efficiency as well. And we are beginning to create a population to do genomic selection. We're still in the very early process of creating the reference population, doing the evaluations there yet. What you see down here is what some of the partners of this breeding program do. This is what we do here at Embrapa headquarters. We do have partners that I will mention to you a little bit further down, and they do pedigree selection, as you can see here. So if you look at these two, are part of the first components that our breeding program has here. This is our backbone. This is where cultivars are produced, and this is where lines are released, and this is where we concentrate much of our efforts. It's important to mention to you before we go into the details of this modified pedigree. Here is where we breeders put art to work. When we say art, we're talking about our capacity to identify plants in the field, but also our capacity to use the correct experimental techniques, the most advanced biotechnological tools, the statistical tools, our breeding knowledge. So it's not only art per se, but it's art in the sense of being able to combine all of this knowledge that we get from the university and put them together to develop a breeding and improve the lines. One thing that we hear quite often is that breeders need to be lucky. Yeah, I agree with that. And breeders, to be able to release cultivars, need to be lucky. There is no doubt about it. But I want to give you my definition of luck before we move on. For me, luck is to work in the field from sunrise to sunset and talk to your plants. Then luck will come to you. If you do that every day along the growing cycle of the plant, along the breeding cycles, then luck will come to you. So yes, breeders have to be luck, but this is the type of luck that I would consider as a main component of a successful breeder. Also, breeding does not happen in the labs. It's a field activity, and it requires a lot of work to be successful. So if you are running breeding programs elsewhere, and you are lucky, I guess you are working 7 to 8 hours in the sunshine, talking to your plants, going to the field, using the best tools. And this is what we do in our breeding program here. As you can see, we get the breeding lines coming out of the recurrent selection program. They come and feed our crossing blocks, but also the elite lines coming from our gene bank, they also feed our crossing blocks. We do about 200, 150, 250, depends on the year, crosses for this specific component of the program. We run breeding nurseries, all this F1s, 2, 3s, and then do family observation, then family building trial. On the components here, we do have time to look at the grain quality, the main components that are demanded by the farmers. And if we are not satisfied with the line, we bring them back, cross them again. If we are happy, we move them forward to the next breeding steps. And also you can bring line from here, from our recurrent selection, and bring them straight to the breeding nurseries because they are good, and so on. If you keep going with with the flow, you will find us releasing cultivars up here. Before we release the cultivar, we do a lot of phenotyping, and we also do on this stage here in farmers field trials, we work on the agronomic traits, trying to evaluate seed density, planting dates, all the agronomic aspects that are demanded by our farmers. Every time that something goes wrong, we move the material back to our crossing blocks and start the process again. From our partners, we move the line they developed into the preliminary yield trial. When we get to the point that the lines are homogeneous and ready to be tested in the farmer's field, when we start multiplying seeds for a possible cultivar release, we do some genotyping to ensure that we have seed quality in our process, and also looking forward to see if our Minister of Agriculture will allow us to use it for registration of our material. Not yet, they are not accepted yet, but they are useful for us to identify our materials out there in the field. Cultivars that are released go back into the gene bank and they become part of our materials. If we go one step further and look at the next step, this is where we do back crossing. What we do back cross for? When we have materials that are good and need like herbicide resistance, I mentioned to you there, this is where we do it. Now we pick our best materials, bring them here, bring the gene we want to introduce. Most of the time we associate them with a marker and do the recurrent selection here as everybody does in different breeding programs. We use the genotyping to ensure that we recover our recurrent parent along the process. Normally for us here in Embrapa, we use this marker for grain quality, which is one of the traits we are breeding for. So this is where, again, materials that come all the way from cultivar release that needs an additional trait comes to this part of the breeding program. And also the lines that are coming out of the back crossing program, they feed in different stages, observational trial, preliminary trial. The modified pedigree, which I mentioned to you, is the backbone of our program, is where we release our cultivars. Next step, the fourth part or fourth component of our breeding program is one that has been is a niche market because it focuses on special types of rice. Is where we breed for the Italian type, the black pericarp type, or Japanese rice type. So it's a straightforward pedigree program. And also when we have something here that needs to be correct or we want to add them something like herbicide resist, we bring them here into the back cross program. We use markers. In this case here, the marker is for aroma. And we do have all this phenotyping for grain quality, insect and disease resistance. And here, grain quality is different than it is here because it's for special rice types. So the grain quality here may vary tremendously from what we consider as grain quality in this part of the program. But again, it's a program that is all integrated, even though if it's a niche program, it's integrating the whole program, and it feeds our gene bank. The materials that come here goes to the gene bank, and that's where we focus when we are trying to develop rice for niche market. We do have another component, the last one, which is a recurrent selection program, but this is special for hybrid rice. We have the story lines, the B lines. Here we combine them. We don't produce hybrids per se. We develop lines that we share with partners, the private sector partners. They do the different components in the hybrid rice program, which is seed production and heterosis study. So we only focus on developing good materials in terms of identifying restorer lines and developing bee lines. We use markers for the cytoplasm, the wild, the WA cytoplasm, which is the one used in the recruiting selection process. Again, we genotype the A and B lines in order to ensure ownership of the materials, knowing what we have in our hands. So this is all about breeding. This is where we do our 100% our breeding program. But moving forward, we also look to potential problems that may affect our rice growers, and Xanthomonas is one of them. We do have a preventive breeding program in combination with the public institution in Panama and using markers. And this is where, as I mentioned to you before, the back cross, the marker here, is where we do this preventive breeding for Xanthomonas. And this is very special and very particular in terms of what we're breeding for, looking at prevent some disease-specific Xanthomonas to get into Brazil. Okay, so this is what we do every single day here at Embrapa, and this is what is providing us the results I'm going to be showing you in the next few slides. Here, what you see is a study that we've done considering when we changed the direction of our program 18 or more than 18, like 21 years ago. And you can see there is a constant increase from there, sorry, and it represents 1.25% increase per year in terms of grain productivity. And if you look at here, the two green circles, they are improved varieties that are out there in the market. And if you look at the lines that we have and the spread and the variability within the lines, we have a huge potential to keep our breeding program moving forward and generating more and more advanced lines with very strong potential to release. If you look at this one is one of the materials that came all the way from here, from the very first group of lines that we selected in 2002. This is a group of lines every year. And this is a group of lines that were part of our breeding advanced trials at that time. So this one came out of the 2002 process. And if you look at here, the more recent ones, look at where we are in terms of variability for productivity. At the same time, we run in the opposite direction in terms of growing cycle. We have been able to decrease our growing cycle year after year in 0.57%. And we also decrease the length of the cycles of our recruit selection program. At the very beginning, when I mentioned to you, we had several breeding populations. We reduced the number of breeding populations. I improved our evaluation strategy, and from 6 years we are down to 3, and we are about now to test the 2 years process. So in order to increase even faster the gains that we have been obtained in the breeding program. Here again are the Czech varieties, and you see that we have a lot of material with shorter cycle, and that's quite important for water management, water saving, and also for disease and so on. So our target is to have early maturing material with a very high yield potential. Here are some examples of what I'm talking about. This is a release, VRS A704. Just to call your attention for a detail here, you find the blue color. Blue color we use in terms of marketing our irrigated material. Everything that's green color is to market our upland materials. So this is the type of yield potential we are getting, 15 tons per hectare. These are the partners. I'll mention them a couple of slides ahead. The whole grain, 67%, which is very, very high. And again, it has lodging tolerance and short or medium cycle in this case. And here is where it's recommended for. If you look at here are the different states in Brazil. This is the Rio Grande do Sul and Santa Catarina are the states in the south of Brazil where most of the irrigated rice is grown. This is another one which is was released a couple of weeks ago, which is Clearfield. Clearfield is a trait that provides resistance to herbicide in rice. Well, today, 80% of the Brazilian area is planted with more than 80%, about 90% already in the south is planted with materials with Clearfield trait. 13 tons, 65% of whole grain, tolerance to lodging, and it's released for the whole, almost the whole country, for the tropical and subtropical regions. Sorry. And here, if you look at the picture, it's a very beautiful picture of rice. Again, moving to upland, see that the name is in green. And here is a picture is, our target is for central pivot, aiming at 9 tons per hectare, which is about half of the cost of production of the irrigated one. Short cycle compared to shorter than the irrigated one, 105 days. And this becomes a part of a system where soybean is produced or other crops, horticultural crops, is part of the rotation with different crops in this central pivot irrigation system. This is another one. This is again a herbicide resistant one. We're about to release it. We're in the very final tests. It's not as important trait as it is the resistance to herbicide is not as important as it is for the irrigated one. But if we look at the system, like I just mentioned to you before, it may become a very advantageous trait to have in our upland rice. This is another one that's coming in 2026. So we do have in the pipeline materials that are very promising and will make a difference in our farmers field. Here is a series of special type of rice. Remember when I showed you in our strategy different components of our breeding program? One component is this special type of rice where we do a pedigree. And here are some of the types of rice. We have aromatic ones for the Italian cuisine, the pericarp, the black pericarp one, or the colored pericarp one. And some of them are just about to be released. We have some red pericarp released already, but we have some that are in the pipeline. Hybrids, as I mentioned to you, in 2010, we released together with CIRAD this hybrid, which was not popular. It did not become as popular as we would like it to be. So we moved to line development strategy instead of producing hybrid, because this was very painful to us in terms of cost, in terms of our capacity to produce hybrids. Again, these are the 5 main traits we're breeding for. Up to now, I focused on the yield potential. Everything that you saw up to here has high yield potential compared to what has been or is being planted right now. Disease resistance, as I mentioned, again, blast is the major problem for our rice, upland rice, and irrigated rice. And our strategy for blast is diversification. We have 100 isolates every year we collect in different farmers' field, and we use them to test against our breeding lines. And here is one of the most popular material for irrigated condition, the IRGA 424, which is resistant to herbicide. And you can see it's very susceptible to different lines, to different isolates. This is the BRS A704. I mentioned to you the picture of it before. And it does have a large difference from Pampera and Catiana, which are materials planted in the south of Brazil now. And it does not have many isolates that are able to attack the BRS A704. But our focus is on diversification in terms of source within the cultivar, but also stimulate the farmers to rotate different cultivars. And here is an example of how we are working on it regarding source. There are 20, if I'm not mistaken, 22 parents involved in this crossing here. 14 of them are bringing resistance to blast. These are some of the source of blast. But again, the most important thing is to keep the variability of the cultivars to the farmer for them to use and cycle them to avoid increase or create a prevalent race in their fields. Okay, we've got a problem here. My slide's not moving anymore. Okay, moving on. Sorry. The whole grain is again, the grain quality is another component, very, very important. And here is another example of how well we're doing regarding breeding our varieties for traits that are required by the market. This is BRS 704, as I mentioned to you. If it has a blue color, is an irrigated variety, and the red line here shows how good it performs when the best conditions are given to it. Provides 65% of whole grain, but the important aspect here is how much how much resistance it has in terms of when we decrease humidity. If we have a delay in harvesting, it can tolerate delay in harvesting without losing quality. Let's move to a component that I consider very, very important in our breeding strategy. Breeding is not only pedigree, any of the recurrent selection or biotechnology tools. Breeding is making sure that you go from the gene bank to the farmer's field. And this is something that we missed ages ago. And when we start looking at it as part of a breeding strategy, it gave us figures like what you can see here in this slide. Considering that Brazil has today, as I mentioned, 1.6, 1.7 million hectares, 33% of this is planted with irrigated varieties coming from Embrapa cultivars, Embrapa bred cultivars. If you look at upland, 21% of the upland cultivars planted in Brazil are coming from Embrapa. Unfortunately, only 56% of the farmers are using improved seeds or improved seeds or quality seeds, or they use seeds they produce themselves. But out of this 56, 33% of the market share belongs to Embrapa, and 21% in upland belongs to Embrapa. But if you look at the future, and our projection is that one of the materials that I mentioned to you, will show the picture to you, which has the clear field trait related to herbicide resistance. According to our projection, we'll reach 30%, only this cultivar will reach 30% of the irrigated market, giving us Embrapa bred cultivars 60% of the irrigated market share. This is our projection according to our market studies and all the studies of the competitors we did. and the potential of the materials in terms of capacity to occupy space that other varieties are occupying right now. Similarly, the study that we did for the upland, we hope to move from 31 to 57% in 3 or 4 more years. And this are the main drivers for us to move from 31 to 20 to 57% of the market share. Again, it's very important for us to do these studies before we release the cultivar and before we move forward in the breeding process. Why I'm saying that? Because before we would release the material in the farmer's field, and we would wait for them to open up their space, not knowing really clear about what they're competing against. And now we have internally created what we call business committee that is part of the breeding strategy. The breeders, before they move forward any line that goes into cultivar, there is a discussion with this committee which focuses on the market, looking what are the breeding lines that would be better suited for running against the competitors we have in the markets. Again, breeders before would do the selection, would go all the way through the end, and then would pass to the next step, the materials, and the seed producers will move the materials forward. Now we are moving one step back, created this committee, and the discussions are all going on earlier in the process. And several cases, the lines are not released because they would compete against materials that are from Embrapa or materials that are better than our material. So it's very easy to say it, but it's very difficult to implement it, because you have to talk to the breeders and make sure they understand why this line is better than the other for the market, and not necessarily better in terms of the trade and the passion that the breeder has for that specific materials. In order to do that, we made a strong link with the seed producers. What do I mean by strong link to the seed producers? Now the seed producers were brought to be part of our program. They finance our program, and they help us to decide what lines we will move forward. We meet with them twice or 3 times a year, show them the results, discuss them, why this or that variety is good or not, or the line has this and that advantages. And they help us to decide what to move forward. In addition, they run trials in their farmers' field with their partners. So the amount of data that we have now to make a decision is much larger than the data we had before. And when the material is good, the farmer, the seed producer already has seed of the material and already knows the material, and he markets it for us. We don't have to market it. They do. As you can see here, there's a list of names. All of these are are seed producers that are working with us in our breeding program. Some of them are active members of the breeding program. Others are just seed producers. But we do have a list of materials and a list of seed producers where you can go to buy seeds. Before, everything would be centered at Embrapa, and then we would tell them where to go. Now, all of these steps are out of our hands and helping us to make impact out there. The green ones, again, are upland materials. The blue ones are also irrigated materials. Again, this color thing is part of the market strategy. Before, you would not know. Now, if you see far away that material has a green label, you know that's upland rice. It has a blue label. It's an irrigated material. And if it has a different color like this ERS 901, it is a special type of rice. So again, this association with seed producers gave us a completely different view than the one we had before. Partners. Again, partners are very important, not only the seed producers, but also partners like the ones I mentioned that you can see in this slide here. They are spread all over the country in different areas. This is where the evaluations are done. And these are the partners that we have. If you look at down here, from this one downwards, they are seed producers, except CRAD. And also the Universidade de Los Andes. But the other ones are seed producers that are part of our breeding strategy. And if you look at this, are the members of the breeding program. We have Embrapa unities in some of these places. Other places are areas where the private sector is running trials by themselves, or universities or state institutions. Altogether, in this network that runs from 1975, we have 45 scientists devoted to work with rice. So I think with this should be my last slide, and I hope that you understand why I start by saying that the Embrapa rice breeding program has a successful story, and we expected it to have an even brighter future because the strategy again has all the components that we dreamed about when we start moving forward in our breeding program. Again, if you need any additional information, here are my email address and my phone. Please feel free to contact me. Again, Carlos, thank you for the opportunity to make this presentation.
Thank you, Elcio, for such a very interesting presentation. And we have already a question in the chat, but I'm tempted to ask the first question. How easy or difficult is for, let's say, international students or international graduates to apply for positions or apply for postdocs at your program?
Okay, thank you for the question, Carlos. We are right now in last year, we prepared a call for international students to come and work with us, not only students, but also people that are from the private sector or different companies that want to come here, join us, or different institutions or universities. We are about to put it out there in the main sites of breeding, where breeding is mentioned. And we are looking forward to have as many students as we can hold them here with us, because the opportunities for interaction are very, very large.
Well, nice to hear. Okay, nice to hear that. Question from Emmanuel Osiegwe from Nigeria. It says, are there any special biotechnologies involved in these programs, aside from what you described? And he listed here like gene therapy, viral vectors, and probably GM traits.
We have a couple of biotechnologists working with us in the program, but mainly looking at the markers or identifying new markers for the important traits for the program. But we don't have anything focusing in different traits. We do have a very experimental work on transgenic rice, but still in a very early stage. I don't want to talk about it more than what I did now, because it's still very early stage. And when you talk about transgenic rice, you have to be careful with the market. But we use the traditional biotech tools, and we focus on markers for useful markers for our breeding program. But the biotechnologists do have their own studies and looking for markers. But again, all focus on supporting the breeding program.
On that line, Fernando is asking, how is the integration of plant pathologies and breeders?
I didn't hear it.
How is the integration between plant pathologists and breeders?
Okay. Yeah, we do have pathologists working with us. Some of the 100 isolates that I mentioned there, they are selected by the plant pathologists. They go out in the field, they collect every year the different isolates, they screen them, they screen the breeding lines that the program has, ensuring that we move forward with the best materials for resistance, mainly for blast. Our main focus is blast. That's why I mentioned I focus on blast, because this is what most of the time of the pathologist is on.
Amanda is asking how do you assess recombination per cycle, and how do you decrease that? I imagine it's the cycle length. I don't know.
How do we access recombination?
Let me see if I can. Amanda, if you want to unmute and ask the question. Go ahead.
Hi, thanks for your talk. I'm just thinking back to the slide where you had kind of two figures on the page, where you had one that was looking at genetic progress over the last years, and then you had decrease in recombination cycle on the hand side. Wondering how you were assessing that.
Okay. We have, instead of working on evaluations at the very, say, line fixed, or not fixed line, but the S02, S03 lines, we are evaluating the S0 plants from the very early stage, and using the high heritable traits to select them, recombine them right away using the male sterility marker on the male sterility materials to shorten the cycle. Before, we would do 1 year for recombination, 1 year for S0 development, S01 development. So we would be 3 years just to get to the S01 evaluation. Now we're doing the S0 right there in the field and using that plant that we select the field to recombine right away. So we're missing the opportunity to evaluate material in the line, but we are speeding up the program and ensuring that gaining time. So we're weighing the material, and our main tool to get that is weighing. We don't go obviously in the S0 and S01. There's not much you can do because there's high level segregation, but we weigh the material, and that's our parameter to move forward the material, the best lines. And again, this, you have to remember, that feeds up our backbone, our backbone breeding program. So I hope that answers your question, and thanks for it, Amanda.
Thank you, Elcio. Again, if you guys have questions, please put them on the chat. Who are Embrapa competitors, Elcio?
There are private sector breeders, companies that are producing rice varieties here. We also, in the south of Brazil, the state institution there is very strong and has a very strong breeding program, as well as in Santa Catarina state has a very strong state program. And as I mentioned, private sector is doing breeding in Brazil, rice breeding in Brazil as well. So these are our competitors. There are not that many. You can count them in your two hands. But it's good because they stimulate us to look for different, to improve our strategies and to be always better.
Right. I recall from my days in the wheat program at Syngenta that the way we made money was to receive a royalty per every bag of seed that was sold wheat. Is that the same case for Embrapa?
No, Embrapa does not have that system yet. Oh, let me back up on that answer, Carlos. When we charge royalties, the royalties go to Embrapa headquarters, and it does not come back to the breeding program. Okay, so what we do now is we decrease the royalty percentage and stimulate people to join the program and to pay for our breeding activities. So let's say that the royalties were 3 to 5% or whatever number you use. If we charge 5% and the whole 5% goes to Embrapa headquarters, and that gets spread to all over the Embrapa's activities and do not benefit the breeding program, the rice breeding program,
Right.
The best strategy is to focus on helping, having seed producers to help us to develop our breeding program. But this has been is under revision now. We are creating a unit that will only deal with the royalties coming from soybean, cotton, maize, from all different crops that we breed for, and that pool of money will come back to the breeding program. So yeah, if you answer the same question next year, the answer will be completely different.
Okay. Can any breeder in the world access the germplasm that Embrapa has developed?
That's a tough question, Carlos. Yes.
That comes from
Theoret-
Fernando, by the way.
Theoretically, yes. In practical terms, is very difficult. The Brazilian system, in terms of get all the bureaucracy behind it, is so slow that does not stimulate people to exchange genetic resources with Brazil. We have been criticizing that. We are members of the international treaty. We know the responsibilities that we have. You can ask for anything in Brazil, you will receive it, but it will take so long that you will ask once and never more. Unfortunately, this is a Brazilian regulation that I don't want to comment on.
Okay. Probably our last question. How has your breeding program addressed the phenotyping challenges related to grain quality, like aroma, starch type, starch content?
Okay, we have a lab here that does all the analysis for the traits that are related to the special types, the aroma, and the grain size, because a lot of the special types have characteristics in terms of grain type different than the one we breed for our normal consumption. We have a lab that does all the studies for us and this work as the same way as the pathologists help us with the disease. There is a quality lab here that help us to select for the different traits. Some of the traits that we don't have the capacity to do it here, we partner with the private sector and they do it for us. So that's the way we deal with the special. But it's a niche market. It's a very small part of the program, which is moving forward quite well.
All right. No, there's one more question that just arrived. Let's make this the last one. Once the varieties are released, can other countries get them or still regulated by the government?
Yeah, as I mentioned, we have this partnership with the seed producers, and in the agreement that we have with them, they have 3 to 4 years, depending on the agreement, and sometimes 5 years, that they are the only ones that can commercialize their material. 3 years after that, the lines are open to anyone. And also, let me make clear one thing. The agreement with the seed producers are to one part of the program. So if they select 10 lines, there's a number of lines that they are allowed to have every year. All the other lines belong to Embrapa, and we can do whatever we want with them, even though if they're financing the program. If there's 100 breeding lines at, say, preliminary trial, they select 10, the 90 left, it belongs to Embrapa, and we can continue doing business with them, continue improving them, and we can release a variety from there without having a specific seed producer being part of that release. So we do have, we run in parallel, we do both things, and the lines are there for anyone.
All right. Well, thank you so much, Elcio, for such an interesting conversation and presentation. The recordings will be available pretty soon. And thanks, Brandon and Tiago, for your help. For next week, this is something for you guys that are attending, take note. The seminar will be on Wednesday. The presenter had a conflict for Thursday. So the seminar is an exception that we do. It's going to be on Wednesday at the same time, and it's going to be about breeding sweet corn for African soils by Tonet Van Goyen from Stark Iris Seed Company in South Africa. So we look forward to have you here. Thank you so much. You guys have a nice rest of the week and a good weekend ahead. Thank you, Elsie, so much, and thanks for everybody that participated.
Thank you to you all. Have a good Easter, and thank you for the opportunity.
Okay.
Abraços, Carlos.
Tchau, tchau. Abraço.
Ciao.